Hello and welcome to video 1 of our WestGrid Summer School June 2020 Bioinformatics session on Wednesday, June 17th. So you've just listened to me talk about a lecture focusing on an introduction to next generation sequencing data and diverse data types. Talked a little bit about transcriptional cis-regulatory data sets and where to find them. And now you are in video 1 where we will initialize our workshop directory and explore data. So the video content, just two simple tasks in this video. The first is to set up our workshop directory, and then the second is to explore the files a little bit. So setting up the workshop directory, go ahead and log into the Cedar Compute cluster with your username at cedar.computecanada.ca. Enter your password. And then we can clear our screen. So you're going to go ahead and enter into the workshop directory. Just really quick, we have a reservation for this course just for today. So if you need to use interactive login node, then you can use these account and reservation information. It'll be active today until 5 p.m. and you'll see where this tag is within our scripts. So we're going to go ahead and switch to our training directory. I can just copy this. And if you look at this training directory, people will hopefully start to create directories here. There are just files, a directory which I've made based on Sherlock, and tools. So once we change into this directory, the important subdirectories to know about. So within files, we have process, raw data, and scripts. Inside of scripts, you will see that we have 3 workshop shell scripts, which we're going to be going through today, and we're going to copy those in a minute. We can go into process, and the output from those scripts can be seen in each of these directories. So if you can't get anything to run to completion today, you can always use these files for visualization or to explore them. And then there may or may not be some raw data in here. There is for 2 samples. I'll probably put the last sample in there after this video. So anyways, we have our workshop directory, and we're going to go ahead and create a new one of our own. We're going to call this one Rosie, which is Sherlock's friend. So within our directory, the first thing that we're going to want to do is we are going to, after we create it, so mkdir makes a new directory, we're going to go ahead and change the permissions on it such that anytime we need to share with a TA or with myself, when you have a problem, maybe something's not running, you need help editing a file, you're going to need to have permissions on this directory. So we can do that if we back up. We'll do chmod user group and world read, write, and execute -r on this new directory Rosie. So today's content, as I said, is just wrapped up into 3 different shell scripts. So we can copy these scripts into our directory. So we will copy those scripts. Just make sure they're all there. One, 2, and 3 into our directory Rosie. And now if we look in our directory, we have these 3 scripts. So we're going to go ahead and take a look at one of these scripts. So this, obviously, you're probably seeing the resemblance here. This is my terminal, so when you're looking at the slides, you're looking at this content, it's going to be mirrored here. So if we look at, let's look at Paul 2. So at the top, we have information about our reservation. So this is the account that would be billed, and your jobs will go straight into a reserved set of compute nodes. We're going to have some mail options. So if you want to enable this, in fact, in order to get the script to run, you're going to need to put a valid email address here, rosie@gmail.com. I'm in the editor Vim, but you can use many different file editors for this purpose. Next, we have the CPU usage. So we're going to use 4 gigabytes of memory per CPU over 8 different CPUs, and we're going to give it 3 hours in total to run. This script will only actually take anywhere from an hour to an hour and a half, maybe 2 hours at the very max if there's something going on with the node, and we're only asking for 1 node. And then lastly, we're going to have an output and standard error files, and so these output and standard error files are just going to be named after the job name, or after the script name and the job ID ending in .out and .error. So again, make sure you change this to be relevant to your own email address. The next part of this script, as we scroll down here, we are looking at the requirements. So we're going to be using 2 different software packages, the first of which is Burroughs-Wheeler Aligner, or BWA, version 0.7.15, the second of which is SamTools, version 1.9. We also are going to use a tool that's not actually on the Cedar Compute cluster. So I've installed this tool into essentially this location here using the Conda package. If you're interested in how that works, you can look in the tools subdirectory, or you can inquire with me. But for now, all you need to know is that by running first this source command and then the activate command, that allows us to use the tool within that environment. Next, we have the genomes that we're going to be using. So here I'm specifying both the genome and the index, although just one of them would be used within this pipeline today. We'll just be using the index, but it's good to know where the genome is. Now, if you're not using the human reference genome, you're going to need to put your own genome here, and you'll need to index it yourself. However, if you have a reference genome which is commonly used, perhaps a well-studied organism, we can just save our changes there. Within the species directory, so cvmfs ref.mujik, or however you say it, genome species, there are many different species and genome versions. We are working in Homo sapiens, but there are many here which will already have the FASTA file and the genome file, which we'll be using, and I'll talk about in just a minute. In fact, there's our segue. So the FASTA file, or FASTA file format, looks like this, where it starts with it's just a text file. It starts with a greater than, has information about the chromosome name, accessions, other information, and then the actual sequence, and then for the entire contig, or in our case, the entire chromosome, and then another delimiter showing you that you've moved on to the second chromosome. Extra information, and then the sequence itself. So we can go ahead and look at one of our BWA indexes, or we can actually just look at this FASTA file. And you can see here's our information. All the human chromosomes just start with a bunch of N's, and then we actually get into the sequence. So not that exciting. These are large files, but it's good to know what format they are in. Now, if you needed to generate an index off your own FASTA file, you would do it using BWA index, and we'll talk a little bit more about the BWA tool in just a moment. Another thing that we can look at is if we want to list out, these are going to be binary files, so we're not going to actually look at their contents, but there's a directory which shows the BWA index, and you can see here they have these 5 index files. And now these index files, these are going to be used by the Burroughs-Wheeler tool, and these index files are essentially a compressed version of the reference genome, which allow for it to be rapidly searched when we're mapping reads against it. That's all you really need to know about the BWA index, and when we refer to it, we're going to refer to it all the way up to this last .fa before having the .amb. So we're just going to refer to what we call the base or the root of the index. Coming back to our script, we're moving down a little bit more. The next thing here is that we're going to have the name. So my name right now is Rosie. Earlier it was Sherlock. I'm going to save that. An ID. This is the polymerase 2 data set. So this is essentially a sample ID. The working directory, which here we're going to make a working directory inside our already current Rosie directory. So see where we are, Rosie, and we're going to create a new directory for this data set. This type of organization is nice because we're going to put all of the data for this sample in a single place. Furthermore, when we refer to these with variables, this allows us to repurpose the same script for different samples very easily. The second section here, we're looking at FASTQ files, which we're going to rename under our standardized essentially sample and ID nomenclature. We're going to have a standardized output name for our output SAM file, our output BAM file. Ignore the max2, it's not covered here. Ignore the peaks in the control, also not covered today. And then we also have a bigwig file. The bigwig file is just a histogram of coverage throughout the genome, which allows it to make nice visualizations when we look at the data in IGV a bit later. The next part of this script is the downloading of data from the internet. So we're taking advantage of the fact that Cedar has its compute nodes connected to the internet. So here we are going to actually download our raw data, and then we're going to change the name of that raw data so that it matches our standardized naming scheme instead of just having this essentially tag from the standardized repository where it's uploaded in the ENCODE project. If you want to explore lots of these data sets to download, you can go to encodedata.org, and for problem set 1, this is actually going to be a requirement. That's it for video 1. We now have essentially our header of our script. We have started by copying these 3 scripts into our workshop directory, and we're now ready to learn about the rest of that script where we map and convert the data.